Oh, filtering in. Okay, without objection, maybe I'll begin my intro. Is this okay? All right, great. Welcome, everyone. It's my great pleasure to introduce Yana Safonova. Please prepare yourself for some attempts to pronounce Russian place names that will not be pretty, because I will not do a good job. So Yana received her BS in Applied Informatics from Lobachevsky State University of Nizhny Novgorod, and in the area of applied informatics, and her thesis was in a hybrid algorithm for solving vertex graph coloring problems. And later she received a master's in applied informatics from the same university with a thesis of design of multi-level algorithm for solving minimum fill-in problem. Later in 2017, received her PhD from St. Petersburg State University, working with Dr. Pavel Pevsner, someone who's spoken in our distinguished lecture series here not many years ago, on application of graph models to bioinformatics analysis of hypervariable biological sequences. That was in 2017. She then started a UCSD data science postdoctoral fellowship at UCSD, which lasted for 2 years. And then she sought out and found and received a fellowship from the AAI, the American Association of Immunology, for computational scientists and immunologists, wherein she worked jointly with Pavel Pevsner and another researcher named Corey Watson. And her research area is computational immunogenomics. And with that, I think I'll just hand it over to Yana. Thank you for being here.
Thank you, Ben. Thank you for the introduction, and thank you everyone for being here. It's a great pleasure to to talk with you today. Yeah, I guess I might skip this because Ben pretty much covered it, just with some pictures. Yeah, my hometown is usually considered as the hardest city to pronounce. It's Nizhny Novgorod, but yeah, it can be complicated. So, and let's move to the interesting stuff. I work on problems in immunogenomics. This is a bridge connecting computer science and immunology. And usually immunology is not viewed as a field where many computational problems emerge. But however, we can show that with proper modeling, there are some interesting problems that we can solve using computer science approaches. So for a moment, let's recall some well-known problem in information theory. It's called problem of random trace reconstruction. We start with a collection of the same sequences or strings, and from each copy of the string, we remove each symbol with a given probability, and it results in so-called random traces. The goal of this problem is to reconstruct the original string T. This problem is very well discussed with respect to the alphabet, distribution of different symbols, and probability Q. However, what we are going to do here is try to reformulate this problem. Let's say that we start not from a single string, but from a set of strings. With this complication, we can come closer to one of real immunology processes. So in this case, formulation will be similar. We create traces from the set of problems, and our goal is to reconstruct the original set of strings. However, to describe one of the real processes, we also need to complicate a probabilistic model for it. Instead of just doing deletions, we also add the possibility to do insertions and substitutions. And again, the final goal is to reconstruct the original string set. So because this model describes one of the immunology processes, we start to look at this and actually publish several papers on this. But before going into this detail, let's also try to understand what this immunology context is, because of this, we can also formulate some of the biological applications. So let's go to look at some biology. Immunology is closely related to studying diseases, and new diseases continue to emerge, some of them re-emerge, and we are very interested in understanding how our own immune system can defend us from all these new emergent threats. Our genome also is limited, is too short to encode all possible defenses. So we need a system that can deal with these unpredictable threats. And this system is called adaptive immune system. One of main agents of adaptive immune system called antibodies. They are proteins that can recognize a specific threat. They are called antigens and neutralize them. And once this complex is built, immune system can recognize it from antibody side and destroy it. That's how our immune system gets rid of viruses, bacteria, and all other diseases. And in order to neutralize very different antigens, we need to have a lot of very different antibodies. And we also discuss what different antibodies are. But that's a big challenge. And actually, we have a very elegant way to deal with this. The fact is that antibodies are not encoded in the genome directly, and they present a result of somatic recombination of special genomic regions called immunoglobulin loci. One of such loci looks like this. It has 55 genes called variable, or V, 27 diversity genes, or D, and 7 J genes, or J, where J stands for joining. Once a cell becomes B cell or cell producing antibody, this loci gets recombined. In other words, the recombination process selects one gene from each of the family and concatenates them together, thus producing a new genomic region called VDJ region, or antibody encoding gene. So here you ask a question, how we generate so many different antibodies if the number of recombinations is relatively small, it's just 9,000. So in fact, this process also does additional things. For example, it can add random insertions and deletions at the VD and DJ joinings, and the resulting number of antibodies is much higher than 9,000. So it's actually estimated like billions of different antibodies. But that's not it. Once antibody binds an antigen, it's labeled by immune system as useful, yet it undergoes through additional process. To make this antibody better, immune system generates multiple mutated copies of this antibody and introduce somatic hypermutations. Hyper because they emerge very quickly and with much higher speed than usual mutations. As a result, a single antibody is turned into an evolutionary tree, and the overall process remains evolution-like process. So the number of resulting antibodies can be even higher than original number of VDJ recombinations. And of course, we are interested in studying such repertoires because they're so different, they can definitely be associated with responses to diseases. And that became possible 12 years ago, where first antibody repertoires were generated. The technology behind this is pretty simple. We just capture VDJ sequences. They are relatively short, so we can cover them using sequencing read. And as a result, we turn antibody sequences into error-prone repertoire sequencing reads, or RIPSeq. They are error-prone because sequencing technologies are not perfect, and we should take this into account when we analyze this data. Ideally, we would like to transform this RIPSeq library into something more appropriate. For example, collapse antibodies corresponding to the same reads corresponding to the same antibody, create the consensus, and forget about errors. So just analyze these sequences. We call this problem repertoire construction problem, and for the first time it was formulated 7 years ago. This is exactly the time when I joined this field, and we were one of the first group publishing a paper about this. So when I joined this field, there were no publicly available RIPSeq dataset, just a few, and they were significantly outdated. So to work in this direction, we started a collaboration with Genentech company, and they generated a private dataset for us that we used for solving this problem. Right now, the situation is completely different. Thousands RIPSeq datasets are available, and it's becoming a routine part of any disease study. So this area is rapidly growing. One thing that we learned by analyzing more and more antibody repertoires that it's not very easy to study them. When you have so many different sequences, the most obvious solution is just to look at overlapping sequences and see how many sequences are shared between two different individuals. However, antibody diversity is even higher than we expected. And usually very few sequences are shared between two different people, even if they are related. And also, these sequences are not that interesting as you might think. Well, we thought that maybe people undergo through the same diseases and then develop identical antibodies, but in fact, these antibodies often represent just frequent VDJ recombinations, ones that are generated very often, and they do not necessarily represent functional antibodies. So it means that we can study antibody repertoire just by comparing sequences, and we need to develop new computational approaches for this. So I've been started thinking about this since my PhD studies and postdoc, and during this period of time, we came up with several open problems that I would like to discuss with you today. First of all, well, we cannot study similarity of antibody sequences, but we still have original VD and J genes that are pretty much shared across many individuals. Maybe we can study them. So can we analyze recombined sequences and predict original genes and see how mutations in them influence antibody responses? This was a work with my advisors, Pavel Pesner at UCSD and Corey Watson at University of Louisville. And we also asked the same question, but in the context of specific diseases, let's say flu and HIV. So that's how we started working with biological groups at Harvard, Yale, and Lahore Institute for Immunology. We can ask the same question about non-human species. For example, cows, they are agriculturally important, so we're interested in preserving their health. And that's how we started to work with USDA. And the same question can be formulated for endangered species, because we're also interested in the immunoglobulin mutations and how these species respond to pathogens, because they might undergo through severe loss of genetic diversity. Well, that was another collaboration with Smithsonian Conservation Institute. And of course, if we look at many different species, we can analyze evolution of immune system. So that's where we can create a bridge with phylogenetic and work with phylogenetic groups like Siavash Mirarab's group at UCSD. And when we analyze more and more species, we start seeing many different types of antibodies in addition to conventional human antibodies. And we are interested in question how these antibodies can be applied for developing antibody drugs and therapies. So these are another questions we're working on with Scripps Institute and other groups at UCSD. Today, I would like to briefly talk about some of these problems, and let's start with more computational problem and come back to the repertoire construction problem. This is a very basic problem that we usually solve when we get a new antibody repertoire data set. Again, we need to combine reads corresponding to identical antibodies, create the consensus, correct errors, and report the sequence with multiplicity. As you see, multiplicities can be different because some antibodies can be present in many copies and some of them are just present by a single copy. So this can be formulated as both error correction problem and a clustering problem, depending what you like to solve. If you're an informatician, it looks like an error correction problem. However, existing approaches do not work really well for this problem, particularly because antibody sequences are derived from the fixed set of regions, and error correction algorithms fail to create a good result here. So we started to working on this problem by looking at examples. For example, in this example, we see 6 antibodies with large abundances and a number of small antibodies. Each antibody will be turned into a set of reads proportional to its original abundance, and these reads will be error prone. But still, we expect that reads corresponding to identical antibodies will be similar. They just unfortunately won't be identical because of errors. However, if we create some form of similarity representation, for example, Hamming graph, we can use Hamming distance because most sequencing errors are mismatches. And then we can represent reads corresponding to identical antibodies as very densely connected graph, almost complete graph. We would like it to be complete, but unfortunately it's not because of errors. And the same is happening with other antibodies and antibody singletons just present by a single sequence. It looks like that we can use this information to identify reads corresponding to identical antibodies and create Hamming graph on all reads from the RIPSEQ library. However, because antibodies can be similar but not identical, we also create a lot of spurious edges corresponding to non-identical antibodies. Nevertheless, we still expect that these dense subgraphs are pretty strong and we can identify them in order to select antibodies. So instead of solving this clustering or error correction problem, we reformulated it as a dense subgraph finding problem. Given a Hamming graph constructed on RIPSEQ reads, find its dense subgraphs and report them as antibody sequences. Well, it's a well-known problem in computer science and there are multiple solutions. We chose one of them that worked well on our data because in our case, time and sources are sensitive matters. We wanted to be solved in a relatively short period of time, let's say several hours. And the idea behind our algorithm was the following. Our Hamming graphs are almost triangulated. So we need to add just very few edges to turn them into triangulated graphs. Why we wanted? Because triangulated graph, for a triangulated graph, we can list its maximal clicks in polynomial time. And clicks in a triangulated graph can be then transformed into dense subgraphs of the original graph by just removing edges that we added into the triangulated graph. So this is a heuristic, but we can use very fast algorithms that are used for triangulation, and it resulted in IG repertoire constructed algorithm. So we showed that this algorithm works much better than other existing algorithms. But when we start accumulating more and more different antibody repertoires, it turned out that our solution is not versatile as we thought. And the fact is that we miss some biological background. We already discussed this a little bit. Antibody repertoire is not just a random set of strings generated from different VDJ recombination. It's a more complicated structure. And in fact, it can be decomposed into so-called clonal lineages, and antibodies inside the same lineage are clonally related. They originate from the same VDJ recombination, and they differ by somatic hypermutations only. So each lineage can be viewed as an evolutionary tree. And you can imagine that these antibodies from the same lineage are pretty close to each other. So because they differ by mutations only. And moreover, this property make look different repertoire, look very, very different from each other. For example, for a healthy repertoire, a healthy donor who doesn't have any disease, lineage sizes are very similar to each other. Well, they still are different, but we don't see any dramatic drop. But in a donor who just received some vaccination or immunization, lineage sizes look completely different. And for example, here you see a result of flu vaccination. What it means? It means that our immune system reacts to immunization and tries to destroy this antigen. And it produces many copies of antibodies that are able to recognize flu and make some lineages look very big. When immune system gets rid of this antigen, it collapses some of the lineages and they become small and it turns back to a picture like this. But at the moment of highest immune response, we see that few clonal lineages are very, very abundant. And arguably, this is the most interesting case because we would like to study disease responses using this method. So what dense subgraph would do in this case? Each lineage is presented by many similar antibodies, different by somatic hypermutations only. So they all will represent a single dense subgraph that will be collapsed and as a result of consensus, we destroy many real diversity. So we don't want to do this. And actually we showed that other tools for repertoire construction also deteriorate for such cases because they apply aggressive techniques for correcting the data. So we decided to address this problem and turn this knowledge about clonal lineages into our advantage. So first of all, we added a step of clonal lineage assignment. We decompose original set of reads into subsets, where each subset is formed by clonally related antibodies. Here we will see both error-free and error-prone reads, and we would like to clean error-prone reads. To do this, we still create Hamming graph because it's a conventional way to represent similarity between sequences. But then instead of finding dense subgraphs, well, it will be pretty much the same dense subgraph, we create the maximum spanning tree on this Hamming graph. And then we expect that error-prone reads corresponding to sequencing errors are random changes. So they don't go anywhere. They don't form long evolutionary passes in this tree, and they will be concentrated in leaves. So in contrast to real mutations that get expanded, and as a result, we will see that they correspond to some long passes, we can identify such erroneous sequences. So here is an example of a single lineage. Ink vertices correspond to sequences that were classified as erroneous, and we can clean them. As a result, we removed a lot of variance and created the cleaned tree. Each vertex in this tree corresponds to a sequence that was classified as putative, so we can report antibody repertoire as a set of sequences from old clean trees from the original data set. And as a bonus, we also get this tree as an output, so we can analyze it and maybe learn something more about somatic hypermutations. Minimum spanning tree is one of the approaches in phylogeny. It's not perfect, but it works pretty well for antibody repertoires because it creates intermediate nodes in the tree, in contrast to classic phylogenetic trees, and we can clearly identify mutations between two vertices connected by an edge. And we can also analyze how these mutations behave. For example, how they correspond to antibody positions. We now have all sequences in the same tree, and we can analyze different positions in these antibodies. For example, position A and position B. And we color vertices according to amino acid at given position. So position A is characterized by a single amino acid, the green one. And it looks like it's very important to have the green amino acid here. Each time it's changed, evolutionary process stops. So it's probably very important to have this green amino acid here and don't change it at all. On the other hand, position B looks very different and looks like it can benefit from getting new mutations. So it starts with a orange one, and then it turns into a green one, and then it turns back. So this is a high mutability position. So clearly position A and B are different from each other, and maybe they have different biological properties. So let's look at this a little bit in more detail. We know a lot about properties of amino acid positions in antibody. We know that antibodies have 3 antigen binding sites, exact places where they contact antigen. And it was hypothesized that antigen binding sites should characterized by high mutability because it's beneficial for them to be mutated and gain better ability to recognize antigen. What is complicated here is that it's not easy to predict antigen binding site just looking at a sequence of antibody, because usually it requires 3-dimensional structure of protein, and this is a very expensive experiment, and it can be done computationally right now. However, there are still methods for predicting antigen binding sites. One of them called finding complementarity determining regions, or CDRs, and there are 3 CDRs in antibody sequence. So if we predict them, CDR1, 2, and 3, then we see that position B falls inside CDR2, and position A falls between CDR1 and CDR2. And actually, position A corresponds to a very structurally important place. So if you change it, you can just destroy antibody protein in general. So it looks like what we observe has biological meanings, but because CDR prediction process is not perfect, it means that sometimes it misses some important positions for predicting antigen binding site, but we can probably refine them if we identify high mutability positions that flank predicted CDR regions, and we can extend them and report them as refined antigen binding site. So we also try to map this information to existing antibody structures, and looks like this process actually refines bounds of antigen binding sites. And this is an important step in design of antibody drugs because they need to know where antigen binding sites are. And it looks like with computational methods, we can actually make this process simpler. But one of the important outcomes is that now we can clean these repertoire sequences and do not care about mutations anymore. We can just work with putative sequences reported at this step and analyze mutations in them as natural variations, not as artificial mutations. So we can come back to the question we formulated at the beginning and analyze variations in V, D, and J genes and see how they influence our antibodies. In fact, something is known about this, although not a lot. And for some antigens and some diseases, it was shown that they have associations with germline V, D, and J genes. For example, influenza or flu is associated with V gene called V169. Moreover, variations in these genes are associated with successes and failures in response to flu, and the same is happening with other diseases. So we need to know variations in the genes and analyze their diversity. It's still a challenging problem because most known immunoglobulin V, D, and J genes come from the European population. And if we use them for analyzing repertoires from other populations, we will do inaccurate inferences. So one of the efforts is to improve the diversity of known genes. On the other hand, we cannot assemble immunoglobulin loci from genomic data because they are highly repetitive. And even modern technologies usually create several substrings of immunoglobulin locus, and sometimes we don't know which part of the locus they correspond to because, again, they are highly repetitive. So that's another trickier part. And even if we assembled immunoglobulin locus, we need to do assignment of immunoglobulin genes, which can be solved. Sometimes it's challenging if this is a new species, but still it's yet another computational problem. So because of all these challenges, bioinformaticians asked, maybe they can use cheap RIPSEQ data instead to analyze the diversity of original VD and J genes. Yeah, it's slightly different. We don't see non-recombined immunoglobulin loci with VD and J genes, but instead we see many rearranged VDJ sequences, and maybe with proper methods, we can reconstruct what genes were used for creating antibodies in this given individual. So this problem is called de novo inference of immunoglobulin genes. And in fact, it was pretty much solved for V and J genes. They are long enough and conservative enough to just perform alignment to the known alleles, identify closest genes, identify differences, apply some criteria saying that these differences are significant, and then report these differences as a novel variant. So that's how the database was expanded. However, for D genes, it's not working because D genes are much shorter than V and J genes. And during VDJ recombination, they also truncated from both sides. And as a result, it's very hard to understand what D gene was used for creating a given VDJ sequence. You can see on this example, we cannot decide because this gene and this gene, they have alignment with the same scores. So alignment doesn't work. We need something else. And that's where we saw these parallels with the trace reconstruction problem because it looks very similar. So we see fragments of original sequences, let's call them traces. They undergo through changes using some probabilistic model that can be pretty complicated one, but still it can be modeled. And our goal is to infer original strings. So we started thinking about it as both theoretical problem and practical problem. And from theoretical sides, we created several models. They are described in this IEEE paper. And the simplest model when these D genes are truncated from the right and extended at the right. It can have an exact solution using computing a trie and maximizing probability to infer specific sequence using it. It also have a greedy fast solution where we just look for the most loaded path in this trie, where each multiplicity corresponds to multiplicity of a prefix. So that looks nice. However, if we also truncate sequences from the start and extend at the beginning, then we cannot apply such data structure. And we also need to create a practical solution for our data that contains millions of sequences. So in addition to this trace reconstruction work, we also developed a tool that can be applied for the real data. Our idea is very simple. Well, we cannot align D genes and say, okay, this sequence corresponds to D gene, but we can identify fragments of antibody definitely containing D gene. We can just cut V gene and J gene, maybe inaccurately, but still. And we also expect that the same D gene will participate in multiple sequences, maybe with different substrings, and we don't know what the substrings are. But if we fix a size of k-mer, let's say 15, that is shorter than any D gene, then we can analyze the most abundant k-mers in the set of strings. And maybe using these k-mers, try to reconstruct original D genes. So that's what we did. And for a single individual, we created such truncated strings, analyzed their 15 mers, and the most abundant 50 mer comes from a known D gene D33. It looks like this. So it's present by 3,000 sequences, and maybe we can use them to create a longer sequence of this gene. So we recruited all sequences containing this 15 mer and tried to extend them to the left. And we noticed that most of them have the same nucleotide. It's actually A. So we can turn this 15 mer into a 16 mer, and this 16 mer is also a substring of the original gene. So the idea is working. And we can repeat this procedure and start. At some point, we cannot extend our sequence anymore because we either reach the start or the end of the gene, or we start seeing random nucleotides. In fact, for this gene, we couldn't reconstruct 3 last nucleotides because they are, in most cases, they are truncated, so we never see them in the data. And our algorithm decided to stop, not to create false inferences. So we call this algorithm IGScout, and it reconstructed all genes with high recombination frequency. And on average, it misses 1 or 2 nucleotides on the left and on the right of the genes, but reconstruct most of the part. We also applied this algorithm for non-human species, and in total, we reconstructed 25 novel D genes for important model organisms, such as mouse, rat, macaque, and others. And now we are adding these genes to the database. We also found that some D genes are associated with diseases such as flu and hepatitis, and we also found that D gene composition differ between mice strains. So it means that the strains should be treated basically as different species because they have different immunoglobulin genes. Right now, they are treated as the same species, and that's create a lot of mix-up in immunology analysis. So that was quite useful, but when we looked at DGens, we also found a very interesting case. We found that some antibodies are formed not from a VDJ recombination, but VDDJ recombination. So two non-overlapping DGens participate in formation of antibody. They're also called tandem fusion of DGens. Well, we were not first, and actually such recombinations were hypothesized by Tanigawa, who discovered the mechanism of VDJ recombinations and got a Nobel Prize for this, and also experimentally confirmed by many groups for the first time in 1989. Unusual recombinations sometimes happen, and in most cases, they are treated just as a curiosity. Well, it's just something that sometimes happens. But in case of tandem fusion, the situation is different because tandem DD fusions result in a longer CDR3 and a longer antigen binding site. And such antibodies are specifically useful against HIV. HIV is a tricky virus. It has a lot of mutation on it, and that's how it escapes from immune response. But using this long antigen binding site, antibodies can target conservative sites buried inside the protein, and that's how they recognize HIV and neutralize it. So basically, mostly neutralizing antibodies against HIV in humans have tandem DD fusions. That's why they represent a certain interest. They even listed in the classic immunobiology book, and it said that although these recombinations are rare, they are important, and we still don't know how they are formed. So we decided that we can try to tackle this problem. We now have a lot of data from many different people. Probably we can learn something about these tandem fusions. And actually what we learned was quite interesting. For example, we showed that usage of DGens in tandem fusions do not correlate with the usage in standard recombinations. Let's say some highly used DGens are never seen in tandem fusions, and some rarely seen DGens form a lot of tandem fusions, so there are some mechanisms behind it. And another feature is that tandem fusions follow the genomic order of DGens. So it means that the first gene in the pair also precedes the second gene in the pair in the genome. And it's actually very important. We know that recombination goes from right to the left, from J genes to D genes and V genes. And using this feature, we showed that this is one more step of VDJ recombination. So it's not something else. It means that recombination just makes one additional step connecting two genes together. So to explain it, we need to learn something about VDJ recombination. One of the important piece of information that immunoglobulin genes are flanked by special sequences called recombination signal sequences, or RSSs. They guide VDJ recombination, and they can be classified into two types, logs and keys. Let's say that V genes and J genes have logs, and D genes have keys, and keys can be only recombined with logs. That's how VDJ recombination is guided. So it goes through J to D and through D to V, and it never performs meaningless recombinations. So DD fusions represent exceptions from this rule, and maybe we need to look at this. Of course, this is a metaphor, and we know the structure of these RSSs. Keys and logs both have a conservative 7-mer separated by either 23 or 12 nucleotide random spacer, and then followed by a conservative 9-mer. So that's the only difference between them. So we can hypothesize that maybe some DGens have key RSSs, and they trick proteins enabling VD gene recombination, and these keys make them look as a V gene or as a J gene, depending on what size they are found. And as a result, we can maybe explain frequent tandem fusion using this hypothesis, just by creating this connection using such keys. So we called such RSS cryptic, and our goal was to find them. Once we found cryptic RSS, we can predict fusions it can produce. For example, if a key is located on the right, then corresponding digit can be recombined with all digits following it, because this key works for all logs on the left. And similarly, if RSS is found on the left, then it can be recombined with all genes preceding it. So we can analyze the genome, find cryptic RSSs, and predict pairs that they can produce. And because we know structure of RSSs, this can be done relatively easy. So in fact, we found cryptic RSSs for some genes, and then we mapped this information to frequent tandem fusions. We explained 95% of observed tandem fusions with pretty good p-value. So we now can say that this is not a random observation. It's indeed connected with cryptic RSSs. And we also found cryptic RSSs in many other mammalian species. And that's why we hypothesize that such TD fusions is not just a random bug of VDJ recombination, it's actually a conservative feature preserved in many mammalian species in order to produce antibodies with long antigen binding sites, maybe to fight something similar to HIV. Actually, some species don't have cryptic RSSs. For example, rabbits, we didn't find any. And that's an irony, because rabbits are used a lot in HIV studies. So right now we can say that if the goal of the study is to find how ultralong antibodies are formed, then we can find a better model organism, because rabbits just don't develop such antibodies. And of course, this is not the end. For now, it's an open problem how to find tandem fusions in highly mutated data. If data is not mutated, then we can find them using some longest substring algorithms. But if you have a lot of mutations, then this algorithm produces many false positives and false negatives. So our next goal is to incorporate information about genes that have cryptic RSSs in order to find tandem fusions with high accuracy. So here we were talking about computational problems, but we also started some work in how to apply them in real biomedical applications. For example, how to make a better vaccine to flu. In fact, the vaccine doesn't work really well, even if the strain was predicted with high accuracy. And to understand this, we need to look into anatomy of flu response. We know that most neutralizing antibodies are derived from the same gene B169, and it has 14 alleles. These alleles can be characterized either by phenylalanine or leucine at position 54. And we know that approximately 15% of the population carry L alleles, so they don't have ability to recognize flu using this gene. For some reason, this gene is also associated with the overall response to flu. So if a person is homozygous by L alleles in this gene, they don't develop good response in general. So other genes, for some reasons, do not replace for V169 at all. And in collaboration with these groups, we started looking at L donors in more details, and we found that they have special set of genes working in response for flu. They just don't work that well as genes in F groups. So our next step is to modify the original vaccine to make it more recognizable for this set of genes. The same can be applied for HIV. DD fusions is one of the approaches. Another approach is to find vaccines that can stimulate antibodies with very fast production. One of such classes called VRCO1, and they also made from gene V12 that is highly polymorphic. And now we showed that the third part of the population carry alleles that don't result in VRCO1 antibodies. So this very common approach that just don't work in 33% of the people. So you can apply similar ideas for study animal responses, and with one goal to preserve their health, and with another goal find interesting features of antibodies that has therapeutic applications. For example, 10% of cow antibodies have ultra-long CDR3s that are even longer than CDR3s resulting from tandem fusions, just because one of the genes in cows is much longer than humans. So again, it was applied for HIV, but we don't know much about the native role. We started working on this problem from the agricultural side with USDA, and they are interested in fighting the bovine respiratory disease. And again, vaccine against it doesn't work well on the population of cows. So here we also found variations in one of the gene and showed that it associated with both loss of ultra-long CDR3s and response to the vaccine. So right now we also know which individuals have a good potential in fighting the disease using genotyping. This idea can be also applied for endangered species such as black-footed ferrets that are susceptible for deadly salvatic plague disease, and there are a lot of efforts to preserve these animals, and then they are released wild, they get disease, and they die from it. So we also identified individuals with good response, and we found what immunoglobulin genes can be associated with this. So I'm running out of time, so let me just make a short summary. Right now, we worked a lot with variations in immunoglobulin genes, but because antibody repertoire is such a complicated subject, there are so many processes happening. We can extend this list of features from mutations to, let's say, structures of evolutionary trees, to 3D architecture of immunoglobulin loci, and other features. That's what I'm planning to do in future. I plan to work on both human and non-human repertoires, integrate other technologies, and hopefully at some point, when we accumulate many associations, we can do data science and machine learning approaches for predicting how individuals will respond to a given vaccine or antigen. And we just started touching a base with flu, HIV, and this species, but I'm pretty sure that in other cases, we can also find interesting associations, for example, with COVID severity or such non-infectious disorders as cancer, autoimmunity, allergy. And our current successes give us ideas that we miss some portion of information about antibody responses, and I'm pretty sure that tools that we develop can help to analyze it. So at the end, I'd like to thank my advisors, Pavel Pevzner at UCSD, Corey Watson at University of Louisville, their labs, and my current collaborators from different parts of the world. And thank you very much for your attention.
Thank you. Great. Questions for Yana?
If you have any questions, I would be happy to take it.
I'll step, if you don't mind, I'll go in. So for the, if I understand your algorithm for the D gene assembly, you're starting with short k-mers, like 15 mers, and then you're doing this kind of one base extension. So it sounds like a classic sort of greedy assembly algorithm, which is normally, there's big risks for, chimeric assemblies or misassemblies. So I'm wondering, what are the strategies to prevent that? And then, furthermore, for these tandem duplications, how you've been able to validate them?
Yeah, so this k-mer extension looks like something that can be used in the de Bruijn graph, but we should keep in mind that this de Bruijn graph will be highly repetitive because the genes, again, are similar to each other. Sometimes a prefix of one gene is a suffix of another gene. So we should somehow deal with this, and we not always have a read spanning through all these repeats. So yeah, I think that can be turned into an alternative approach. It's not just, it can be applied directly and it produce some results. So we did some tricks here to make it working. For the Bruijn graph, we probably need that too. But in general, yeah, there are some similarities. For example, here we have a problem if we have a allelic variation. Let's say that we have not, where do you have this? Yeah, let's say that we have not a dominant nucleotide but two dominant nucleotides, which is possible because of natural genomic variation. Our algorithm actually has a lot of difficulties. Maybe an alternative approach will help here.
And then kind of to follow up, where do you see the field moving? Is it towards, maybe hi-fi reads that, can be longer with higher accuracy? Is it, totally new algorithms, a different type of data? You tell me what would be most informative.
So when we work with recombined data, we already sequenced what we can. It's just that these genes can be always truncated, and we don't have any VDJ sequence, so we can span the whole sequence. So that's why hi-fi reads for this particular problem won't be that useful. If you want to analyze original loci that are not recombined, that's certainly a solution. But then we need to do assembly, make sure that it's working and that we can predict digits there. For humans, I guess it's pretty much can be solved. And we also see very conservative haplotypes of digits. So maybe for this problem, we already did what we need, and we just need to do it for other species right now. Yeah, so hi-fi would be useful for assembly. You also asked about tandem fusions, how we validated them. So first of all, we developed approach. I didn't have time to talk about this in details, but we developed algorithm for finding cryptic occurrences of conservative 7 mers and 9 mers in the genome. And we looked specifically at immunoglobulin locus, and where we found the most conservative 9 mers, that's actually where cryptic occurrences of such key RSSs indigenous. So we just there applied statistical method to say that what we see is not random, because the lower threshold for detecting RSSs, the more data we start to capture, and at some point we don't know what data can participate in such cryptic recombination and what cannot.
Right.
Yeah, so we also looked at some validations from experimental papers. They actually showed that such double RSSs can work. Yeah, it was a popular subject in 90s when mechanisms were just discovered. People start seeing how they exactly work and how differences in RSS signals change the activity of proteins, how they can work outside immunoglobulin loci. So we try to also explain this in these terms. And here we again can create connection with hi-fi data because they are longer, and sometimes standard Illumina doesn't capture long enough antibodies.
Right.
Yeah, so here it can be interesting to do just to see what kind of diversity we currently miss.
Thank you very much.
Thank you.
More questions for Yana?
Hello. So
Hello.
great talk, and I wonder, have you tried using like whole genome sequencing data to analyze the like populational mutability for like Ig or T cell genes? Because I think the datasets are rich, and I know they are before recombination, so that might not like perfectly like characterize the Ig characteristics, but we have lots of data there. So have you think about that, or have you tried that in your previous works? Thank you.
Yeah, thank you. That's a great question, and that's actually this is a big hole in our field. Well, one of the sources of such data is one key project that tries to capture as many diversity as it can, but it results in pretty short Illumina reads. And the problem for assigning this reads to one of the immunoglobulin genes is pretty big because genes are similar to each other. So at some point you need to make a decision or just throw this read away. So there were several papers that tried to do this, but unfortunately it was shown that false positive rate for them was relatively high. So for Illumina, I would say that it's not easy to do. With PacBio data, it's getting more realistic because they are long enough, and if again we work with hi-fi reads, and they are pretty accurate. So there are such efforts, and yeah, ideally, I would say that it would be nice to do both genomic data, because sometimes we cannot infer all immunoglobulin genes and T cell genes which are in a similar situation. But using genomic data, we sometimes cannot just place a single read to the appropriate location in the locus because of high repeativity on this locus. There are basically 5 long, very diverse duplications in the IG locus. And when we have a single read, we don't know if it comes from this part or that part. So we don't know what reads we need to assign it to. And ideally, we would like to do whole genome sequence, assemble individual IGH locus, and then also have repertoire seq data to see which genes are expressed and how they're expressed. So such data sets are currently unavailable, and they also have challenges from all sides, but this is a plan. Okay.
Thank you.
I have a quick question, which was back at the discussion of the RepSeq and sequencing repertoires. I wondered if you've thought much about the time dimension as well. Like, for example, you were saying that the spanning tree approach, I think you were saying that one of the advantages is that the internal nodes of the spanning tree actually correspond to items in the repertoire, as opposed to being like hypothetical. I think I lost you. Okay, sorry, my internet connection is a little unreliable. But so I wondered if, therefore, I guess the assumption is that the more mutated items in the repertoire are not replacing the less mutated versions; they're all coexisting. But is there a reason to think that, or could it be that the more mutated sequences are actually crowding out and that the other sequences are actually degraded and therefore shouldn't be in the tree?
Yeah, that's the problem with the maximum spanning tree. We observe both. Sometimes we see that both ancestor and their descendant are present, but sometimes we see that only descendant are observed. And this happens, for example, with pretty mature responses. For example, if you look at a person infected with HIV, then most of the intermediate antibodies are lost and we observe only far relatives. So if this is true, then we can try to apply phylogenetic approaches because we don't need to care about intermediate nodes. In most cases, we see a mixture, and the maximum spanning tree has its own problems. In case of missing ancestors, it will just put edge between siblings, so we can miss directions between mutations. So we are aware of these problems and we like to address them in future. Right now, we just try to make conclusions that do not take into account directions between mutations because we know that they are inaccurate. Yeah, so this is just very simple. And once we create a set of accurate sequences, we can say, okay, this tree just show us something. Let's try to make a good tree from it and then apply additional phylogeny or evolutionary approach to recreating better tree for this.
Thank you. We have maybe time for one last quick question. Okay, if not, then let's please thank Yana again. And thanks very much. And everyone can rush off to their 3 o'clock Zoom. Thank you, Yana. Thanks, everyone.
Thank you.